Assalamu alaikum dear students. So now we will discuss DNA in more detail. So here is the nitrogenous bases are present, these are nucleotides. So we say these two strands of DNA are separated from each other by the distance of 20 angstrom. Here is the distance between the two strands of DNA, that is 20 angstrom distance between two strands. The diameter of DNA is 2 nanometer. The gap between two nitrogenous bases is 0.34 nanometer and 3.4 angstrom. The gap between the 10 nitrogenous bases is 3.4 nanometer and 34 angstrom. These are if we want to know the distance between nitrogenous base, or we want to know the number of nucleotides in any DNA strands, you must keep in your mind that the number of DNA in the DNA number of adenine and thymine always remains same. If a DNA molecule has 500 adenine, so it is must there is the number of thymine also 500. If the number of cytosine in DNA 400, then guanine is also 400. If we want to know, there is a DNA molecule that have 1000 nitrogenous bases. Out of these 1000 nitrogenous base, that have 200 guanine molecules. Total nitrogenous base total 1000, and that total nitrogenous bases, and that has guanine 200. So we know the number of guanine is 200, then cytosine is also 200. So total number of DNA nitrogenous base guanine and cytosine is, this is cytosine, guanine is 400. So total number of DNA, total nitrogenous base present in DNA molecule are 1000. So we minus 400 from 1000, then the remaining is 600. And when we divide 600 with 2, so we obtain 300 answer. So in this way we can find the number of adenine or thymine. We can trace easily number of thymine or guanine or unknown strand from the DNA molecules. In this way we trace, there is total number of nitrogen bases given and one nitrogen base is given. So that guanine, the amount of guanine is given. So we can easily trace the rest nitrogenous bases number. And if you want to know the number distance, there are 100 nitrogenous bases present in the DNA molecule, and